Hello, everyone. My name is Hamidreza Mahyar. I have PhD in computer science, and I was postdoc researcher at Boston University, all in the field of machine learning over graph structured data, like social networks, recommender systems, or even protein-protein interactions. Today, I will be talking about the work done by some of my colleagues here at ProteinQure and focusing on our protein design with deep generative models. What are proteins? Proteins are molecular machines that exist in all living things, which play a role to keep us alive and healthy. At a very high level, our cells read our genetic code, let's say DNA, and convert that to protein sequences. These protein sequences fold into a 3D shape, and because of that shape, they can achieve some specific functions in the body, like fighting a bacterial infection, for example. With advances in technology, scientists have been able to produce new proteins that exist outside of nature for diverse applications across industries, including pharmaceutical research, which is what we focus on at ProteinQure and what we will be talking about today. One major issue with this process is that it can still take years of trial and error to design a protein for any of these applications. So for AI audience with limited biology background, let me abstract the problem this way. In nature, we have 20 natural amino acids that are building blocks of a protein. They're depicted on the right-hand side. As you can see, we can have one alphabet for each amino acid. A protein from a sequence level point of view can be considered as a sequence of variable lengths where in each spot or position, one of these amino acids can sit. Overall, for a sequence of length n, we can construct 20 to the power of n different sequences or proteins. As you can see, we have very large sequence space, and we have a lot of complexity there. And among all of them, only some of them have our desired properties. So how can we ever hope to perform design? Our goal at ProteinQure in general is to design protein therapeutics, shown as orange here. And for that, we need to find very specific proteins that stick or bind to their disease target, which is shown as green here. One advantage that we have is that we know a lot of sequences of natural proteins that have interesting properties. So how can we use that information to help design good drugs? I'll explain how we did it for one of our recent projects with one of our pharma collaborators. With the help of several computational biology tools, we designed 3 protein libraries containing sequences of amino acids that with high probability, they tend to bind to our target. And these proteins were used for training a deep generative neural network model in order to generate novel and diverse set of sequences. And besides that, we also used some public data set, but this was our inside data set that we developed. As a high level, this is a graphical depiction of the process. Assume there are 3 different clusters of sequences that are share common properties, blue, green, and red. An example sequence in the blue cluster is this sequence on the top left hand, which is HN, WR, and YE, where HN and YE on the 2 end are the variable sites, and the 2 amino acids in the middle, WR, which stands for tryptophan and arginine, is the constant binding motif. The generative neural network learns the distribution of these different clusters depicted by these three circles. It then tries to combine different behaviors of different distributions to generate new sequences with combined properties. For example, this magenta sequence on the top right has been generated as the result of combining different properties of blue and red distribution, and exactly the same for orange sequence. For our neural network architecture, we decided to use a Wasserstein generative adversarial neural network model with gradient penalty. Basic schematic of a Wasserstein GAN architecture is shown here. The generator and discriminator are both parameterized as deep neural networks, and I will show you the architecture in the next slide. But in this architecture, real data is encoded to a one-hot vector, and the generator learns a continuous approximation to this encoding. For more technical people, you might know why we used Wasserstein GAN instead of the original GAN, and why we used a gradient penalty instead of weight clipping to enforce Lipschitz continuity. But I don't dig into this technical detail. For the general and detailed neural network architecture that we used for generator and discriminator, you can see that, for example, for generator, we take the random variable, latent variable Z as the input. We have several layers containing the linear layer, and a couple of residual blocks with skip connections. We have the convolution 1D, and then we have the softmax, and the output would be a sequence. And on the other hand, on the discriminator side, we take the batch of generated sequences and the real sequences as the input. And then we have somehow the reverse architecture as the generator. We pass them to convolution, residual block, and then linear, which gives us a score in order to decide whether the generated sequence is good enough or not. And we trained this huge Wasserstein GAN with gradient penalty over around 500,000 iterations. But as you can see in the learning curve of training set and validation set, after around 200,000 iterations, the model could converge and could learn the distribution of the data. In order to test whether the generated sequences are good or not, we, for example, first test the probability density estimation of training, validation, and generated sequences. And as shown here, the generated sequences are diverse, yet holding the desired property, for example, the hydrophobicity. The lower the hydrophobicity is, the better sequence will be. As you can see, the hydrophobicity of training set is ranging from minus 0.8 to 1, but the GAN-generated sequences have hydrophobicity ranging from minus 1.2 up to 0, for example. But as another test to make sure that our model didn't overfit to the training set, we check the edit distance between the around 20 randomly sampled data as a test set to the training set, and the edit distance between the generated sequences to the training set. One clear observation is that both the generated and the test sequences are concentrated around the same distribution. Let's say the edit distance 7. But the generated sequences are on average further from the training sequences than the test set, which indicates that the model has not overfit. So overall, we use generative adversarial neural network to design protein therapeutics. We were successful in training a model to generate diverse protein sequences with desired drug-like properties. We use this model to construct a protein library which is currently being experimentally evaluated in cell-based assay with our collaborator. Our ongoing work will improve scoring accuracy through feedback cycles with new experimental data. And for anyone who is more interested to the technical detail, it's worth mentioning that we also tried, for example, recurrent neural networks or variational autoencoders, but we didn't get good result at the end. That's why we decided to use generative adversarial neural network. So thank you all for listening to the presentation. Please check our website for news, updates, and opportunities and all the openings that we have.